In Chapter 8, we are going to delve into the details of how DNA is used to be copied, and then we're going to look at how it's used to make a protein. We introduced DNA, RNA, and then how we string together amino acids and to make our proteins back in Chapter 4. If you need to go back and review base pairing information or structurally what DNA and RNA are, how they differ, make sure to go back and rewatch some of those explanations so that you can refresh that information if you need to go back and do so. Today in lecture video A, we are going to cover slides 1 through 10. Lecture B will cover 11 through 14, and then lecture video C is going to cover slides 15 through 22. So let's begin. All right, we're going to start by talking about bacterial genomes. So the term genome is a broad term that relates to all of the DNA within a cell. So we are focused on prokaryotic bacterial cells. So up till now, we have really focused primarily on bacterial chromosomes. We have said that a chromosome is a long stretch of double-stranded helical DNA. We said that that DNA had base pairing across the helix where A's paired with T's and C's with G's. In the vast majority of our bacteria, their chromosome is going to be a circular piece of that DNA. Now, there are some that are linear, but in general, they're going to be circular. On this big loop of chromosome, we have individual segments that are what we refer to as a gene. So a gene is a short stretch of DNA on this larger loop of chromosome. A gene is a unit of information in that the gene, the sequence of nucleotides on that little stretch of the chromosome, is going to be the information that is needed to make an RNA molecule and ultimately make a functional protein. So when we start talking about the different genes that one species have versus another species, what we're really talking about are the genes that are present on that big chromosome. So those chromosomal genes are going to dictate the enzymes that can be made, and therefore it's going to dictate the metabolic processes that a cell can do. When we talk about different structural components that may or not be present with certain species, well, those pieces of information, those instructions are going to be found on the genes within that chromosome. But up till now, that's really the only source of DNA that we've talked about. So I've misled you just a little bit. Bacterial genomes are more than just that chromosomal DNA. Bacteria are special because they have additional small loops of DNA that are considered to be extra chromosomal, meaning that they're kind of fun pieces of DNA. They don't have the necessary genetic information, but they can certainly be important. Oftentimes when we have characteristics that are unique to a particular subset of a species, they may be found, those genes for those characteristics may be found on these plasmids. So this image up here is one that I've pulled from Google because your book just doesn't have a good representation of what this kind of looks like. So here we see our bacterial chromosome with all of those critical genes that make that cell part of a larger species. But we can have these extra little loops. There's oftentimes more than just one plasmid. And it's on these plasmids that we see really kind of non-essential genes. So these may be things for, like I said, extra virulence. So when we start talking about toxins that may be produced, well, they could be part of the plasmid, that plasmid loop of DNA. Antibiotic resistance genes are oftentimes found on plasmids. So these are genes that are beneficial for the bacteria to have and give it an advantage in its environment, but they don't necessarily mean that that particular cell is going to be part of a given species. So we have talked a little bit about how we use DNA. We've said that DNA is going to be used to make a protein. That protein is going to ultimately be used for some function within the cell. Well, we don't directly use that DNA. So when we start talking about what's called gene expression, that's this process of taking a segment of our DNA, a gene, and ultimately making a protein. Remember that we use that intermediate molecule RNA. Well, we have not yet specified that there are different types of RNA, but in this chapter we will learn about 2 of the different types of RNA. So now we need to begin specifying which type we're using. In gene expression, where we use a sequence of DNA within our gene and ultimately make RNA and then a protein, the type of RNA that we're going to be making is called messenger RNA. And we denote messenger RNA by having a little M at the beginning of it. All right, so let's then rehash how we need to copy our DNA. So we said that essentially there are 2 ways that we're going to use it. We want to utilize those instructions to make the stuff that the cell needs, but we need to copy that DNA before we undergo binary fission because it's important to pass on the instructions to each of those 2 daughter cells that are going to be made. So before we undergo binary fission, we have to replicate that DNA. Do you remember which enzyme is in charge of replicating the DNA? DNA polymerase is going to be the enzyme that reads our DNA and makes an additional copy. The deal here is that while we like to think that every single nucleotide is going to be copied perfectly through base pairing, DNA polymerase is not perfect. We estimate that DNA polymerase in bacterial cells is going to make a mistake about 1 every 10 million base pairs that it reads. That mistake, that change in the DNA sequence is called a mutation. Now, oftentimes students come in with the idea that a mutation is bad, and it certainly can be. But sometimes mutations don't have any effect on the protein that will ultimately be produced. Sometimes they can even be beneficial. So when we start talking about increased virulence or a change in susceptibility to a given antibiotic, well, those types of mutations, those types of changes in the DNA can actually be beneficial to our microbes. So we'll talk more about mutation and what it means in terms of kind of a larger picture in a little bit, but I want to make sure that you understand a mutation or a change in the DNA sequence isn't necessarily going to be a bad thing. All right, so let's kind of rehash what we know about the genetic code. We said that we need to use our DNA in 2 ways. First, we have to be able to copy it so that when we undergo binary fission, both daughter cells have the correct genetic information. DNA polymerase is going to be the enzyme that does this. DNA polymerase is going to copy all of the genetic information in that cell. That includes our chromosomal genes and those plasmid genes as well. So once we copy and have that information in our 2 daughter cells, well, we need to be able to utilize that DNA. DNA is going to be read by RNA polymerase. RNA polymerase is going to make a messenger RNA molecule. That messenger RNA molecule is going to be read 3 nucleotides at a time, And we are going to have ribosomes that do that reading of our messenger RNA. Our ribosome is going to read those 3 nucleotides at a time, and it will string together the correct amino acid sequence to make our protein. We talked about that back in chapter 4. If you need to go refresh, bring that video back up and go through those slides once again. All right, so let's talk about this DNA replication, copying our DNA. We're going to add some details to what we already know. So when we talk about DNA replication in our bacterial cells, how RNA polymerase is going to copy our DNA, well, that process that DNA polymerase is going to execute is called semi-conservative replication. Semi-conservative replication refers to the fact that DNA consists of 2 strands, and we're going to separate those 2 strands. And then each of those is going to be used as a parental strand. And we're going to synthesize 1 new strand for each of those parental strands. So that's what we have depicted here. And again, your book doesn't have a simple diagram of this, so I pulled this one off of Google. So here's our initial short sequence. This is just part of our DNA. It's double-stranded, and it would be twisted in that helix. We see base pairing across that helix. So what happens is we break the bonds between these 2 strands, and we separate them. We then have DNA polymerase come in and look at this A and go, oh, well, I need a T. And then we look at this G, and DNA polymerase goes, oh, need a C, then a T, and a G, and an A. And what DNA polymerase is going to do, according to base pairing rules, is synthesize this new strand. So this newly synthesized strand in this particular sequence is depicted in red. So now we have 1 copy. Our other parental strand down here, DNA polymerase sees a T and it puts an A, sees a C and it puts a G, a T and it puts an A, so on. And we have this newly synthesized strand down here. This is called semi-conservative because 1 of each parental strands is conserved. It remains in the subsequent copies. The opposite of this that doesn't happen would be called conservative replication, where we would have 2 old red strands, or excuse me, 2 old blue strands and 2 new red strands. That would be conserved, where we have both parental strands in one and then 2 completely new strands in the other. But that's not how it works. That's just an explanation to help you understand the semi-conservative nature of this. So ultimately, once we get this copying done, well, look at this sequence as a whole. It's identical to this sequence down here. And both of these strands are identical in sequence to what we started with. So back in chapter 4, when I asked you to do base pairing, essentially what you did was play the role of a DNA polymerase enzyme. You read what was there, and according to base pairing rules, you copied that, that strand. We've talked in general terms briefly about how this replication begins. We said that it starts at the ORI, which stands for the origin of replication. So the origin of replication is where DNA polymerase is going to have those strands separated so that it can access the nucleotides on each of those parental strands. Before DNA polymerase can act, we need another enzyme to come in and break the bonds between our 2 strands. The enzyme that unzips our unzips our gene is called helicase. So it's an enzyme, it ends in ASE, and it's going to essentially unwind and break apart that helix. By doing so, helicase gives DNA polymerase access to that parental strand. So DNA replication is not only semi-conservative, where we have two strands that are ultimately going to be made, where one strand is a parental strand and another strand is newly made. We're going to have this process starting at that origin of replication, and it is going to move in opposite directions. So it is bidirectional. DNA replication. I like this image because it's nice and it's color-coded and it's simplistic. So here we have our circular chromosome, okay? We have our origin that is depicted here. And so what we're going to do is we're going to have our replication fork here and another replication fork here. We're going to work in opposite directions from that origin. We'll have two helicase enzymes working to unzip in both directions, and we'll have a total of 4 DNA polymerase molecules at the same time, all undergoing that semi-conservative replication. All right, so we're going to jump back to this genetic code kind of shorthand. We have talked now about DNA replication. We said that helicase is our enzyme that unzips, it breaks apart that helix. DNA polymerase is going to execute our base pairing, which leads to semi-conservative replication. That semi-conservative replication is going to move bidirectionally, and we'll just keep going until we reach the end of that chromosome. We are going to begin looking at the big picture of gene expression. So how do we use our DNA to ultimately make a protein? We are going to see that the process of RNA polymerase reading DNA and producing messenger RNA is called transcription. So DNA will be transcribed into messenger RNA by RNA polymerase. When that ribosome reads the messenger RNA 3 nucleotides at a time, that is translation. So ribosomes translate messenger RNA into a protein. We will talk more about the details of these two separate processes in videos B and C, but we're going to kind of keep it a nice big broad picture, what's going on and why we're so interested in the sequence of DNA. The real reason behind it is because those DNA nucleotides, that sequence of our DNA, that gene is going to ultimately dictate the protein that is formed. And so when we start looking at how we do this, we're going to look at kind of a version of what I just showed you but turned on its side. So here we have our big, long, double-stranded helical DNA, and we're going to look at this little short sequence of DNA. This is a tiny little gene. We're going to keep it simple. Well, we look at one of our strands of DNA. According to base pairing, we can make that into RNA, and that's what happens. That's what transcription ultimately is. We've said that we read those nucleotides 3 at a time. We're going to now give that set of 3 nucleotides a name. We're going to call those 3 messenger RNA nucleotides a codon. So codons are what ultimately are responsible for an amino acid. Well, if you change your DNA sequence, if there's one of these letters that changes, well, that's going to affect the RNA sequence. because our RNA polymerase is just simply going to look at the DNA that's there, and it's going to make that RNA, that messenger RNA, accordingly. Well, so if we have that RNA nucleotide that's different, well, that's going to affect the codon, that set of 3 as a whole, and it could potentially change the amino acid. Changing the amino acid would change the protein. So this is how a mutation in our DNA can persist through gene expression and can manifest as a change in the protein that is made. So we will look at this process in detail in a little bit, but we need to understand how this process works at kind of a larger level. So this diagram over here is a diagram that represents the codons that are responsible for each of our 20 different amino acids. So you'll notice that some codons, there can be multiple codons that are responsible for a given amino acid. I don't expect you to memorize any of these amino acids in particular, but I want you to understand that a given codon, so like the UUU codon, for instance, will always cause a ribosome to insert the phenylalanine amino acid. Well, the codon UUC will also result in phenylalanine. Leucine can be inserted based on a total of 6 different codons. I don't care that you can actually memorize any of these, but there are a few of them that I want to point out to you. We will practice using this code in a little while, but one of the codons that is special is the AUG start codon. That one you do need to know. AUG is the codon that codes for methionine, and it is responsible for starting all of our different proteins. AUG will be at the beginning of every messenger RNA that's made. That's how ribosomes know where to start. Well, if they need to have a place to start, they also have a place that they need to end. The 3 stop codons are depicted over here in red. UAA, UAG, and UGA do not code for any different amino acids. They are stop codons. When a ribosome gets to these codons, that's how it knows the protein is finished. So we'll talk more about how we use this in a little bit. You will be expected to be able to use it if I provide you the table on your exam. But right now, understanding methionine is our start codon. We read those codons 3 at a time, and each codon always codes for its particular amino acid. And then we have those 3 stop codons that don't code for an amino acid, but tell the ribosome it's done. Okay? So we're going to look at this process, this genetic code, really in the context of mutations. So how does a change in the DNA ultimately change a protein? Well, we said that there were some instances that it might not. How does that work? Okay, so by looking at the different types of mutations, the different types of changes to our DNA, we can look at different classes of their results. So how does this ultimately influence the protein that is going to be made? So first, let's look at a mutation called a base substitution mutation. So this is where we have one single nucleotide change in our DNA. So we've had a change to the DNA. That is a mutation. Sometimes a base substitution can lead to a silent class of mutation. So a silent mutation is where our nucleotide change doesn't ultimately change the amino acid that the ribosome is going to put in that sequence. Let's look at an example of this. So let's go back to phenylalanine. We said, well, our phenylalanine amino acid could be put there if we have the UUU codon or if we have the UUC codon. Well, in this particular instance, a base substitution at that last position would not change the amino acid, and the protein would be made just like normal. So that would be an example of a silent mutation. Shh, nobody knows that it happened. A base substitution could also lead to what is called a missense mutation. So here we change that codon and change the amino acid. We ultimately change the protein. Oops, I went the wrong direction. So let's say instead of UUC, now our codon is UUA. It's still a base substitution, but now by changing just this one codon, we've changed the whole base. That would ultimately change the sequence and could change the protein. So that would be a missense mutation. We could have a nonsense mutation that takes place. A nonsense mutation is where we have a base substitution or some other mutation where we have changed our codon to a stop codon. A nonsense mutation is going to stop translation early. So not only are we not going to, have just the wrong amino acid, we're going to prematurely terminate translation. So we're going to end up with a much shorter protein than we would need. So nonsense mutation will very likely result in a non-functioning protein, and we'll see that manifested. So the next type of mutation could be an insertion or deletion. When we have an insertion or deletion, we see what is called a frame shift mutation. Frame shift mutations cause a whole bunch of amino acids to be incorrect. When we insert or remove one of our nucleotides or even a couple of nucleotides, we change which group of 3 our ribosome is going to be reading. So we could potentially, if we lose this U at the beginning, well, then our reading frame would be UCA, CUC. We have changed almost every amino acid, and we will have a major change in the amino acid sequence. Ultimately, it will, it will very likely result in a non-functioning protein. An inversion is another type of mutation. This is where we have a segment of nucleotides. They're all still there and they're in the correct sequence, but they have been cut out and flipped around. So this can have varying degrees of impact on the ultimate outcome, the ultimate protein that is produced. So talking through that I know can be a little bit confusing. Let's watch an animation to help us clear up these different classes of mutations.
DNA rarely makes mistakes, but it can happen. A point mutation changes a single base. This can change the whole amino acid, or, depending on its position, could go unnoticed. Other mutations can be more damaging. A deletion removes a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the deletion, the amino acid sequence will change because the bases shift by 1. The resulting protein will likely be non-functional. An insertion adds a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the insertion, the amino acid sequence will change because the bases shift by 1. Again, the resulting protein will likely be non-functional. Lastly, an inversion will invert a segment of DNA end to end. Watch this section closely. Each base has been moved around. Again, these mutations have the potential to be harmful, but not all mutations have an effect on protein. Remember, during translation, a start codon must first be read to begin coding the protein. If the mutation happens before or after the protein coding sequence, there will be no effect on the protein. Mutations must occur in the coding section of DNA to affect protein synthesis. Otherwise, nothing happens. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
All right, so let's keep moving on. All right, so when we talk about mutation, we said that it could potentially not be an issue at all. It may not manifest as a change in the protein. But there are times where, there certainly are times where a mutation could be good. It could change a protein in such a way that it would benefit the bacterial cells. But in general, we're going to see that most mutations are harmful to some extent. And so cells have ways to recognize when mutations have occurred, and they have enzymes to go in and fix those mutations. Because again, in general, mutations are going to have a negative outcome. So there are several classes of mutation repair that bacterial cells can do. The first is called base excision repair. Recall that a DNA nucleotide consists of a deoxyribose sugar, a phosphate group attached to that, and then one of 4 nitrogenous bases. And the nucleotide as a whole is named for that nitrogenous base. So it could be an A, a T, a C, or a G. Well, when this nitrogenous base is damaged, or it is just chemically, it is messed up in some way, it can affect how the DNA is read. And so the cell wants to go in, identify that it's there, and pull it out. So DNA polymerase is the enzyme that is in charge of this. This is what is considered to be a proofreading mechanism, where DNA polymerase is going to essentially check its work. It's going to proofread things and ensure that the sequence is correct, and it's using nucleotides that are appropriate, that are formed correctly. DNA polymerase, we said, was not 100% efficient. So a mismatch repair can occur when DNA polymerase puts in an entirely incorrect nucleotide. So we have to take out that whole nucleotide base. An enzyme is going to do that, but it's not just going to be that individual base. We're going to take out a little bit of information on either side of that. So I kind of like to think of this as sometimes if you go through and you're thinking one thing and you're like typing up an email maybe, and your words don't come out quite right, rather than just going in and fixing a single word, you delete the whole sentence and you rework it. That's a little bit like mismatch repair. And DNA polymerase is not going to be the enzyme that's going to remove that whole big stretch. But once it is gone, DNA polymerase will come in and be like, oh, my bad, I've got to go in and fix that. And it will correctly insert the right nucleotides, the right bases in that particular sequence. So when things go really wrong, oftentimes this is when bacterial cells are running out of nutrients in their environment, energy supplies are low within their cell, DNA polymerase just gets sloppy. It doesn't have the right amount of energy that it needs. Sometimes it doesn't have all of the nucleotides that it's going to need to do this base pairing. And so sometimes it begins to screw things up really bad. So we can go in and do what is called SOS repair. And SOS stands for save our ship. So like if you've ever heard of somebody like, oh yeah, they called out SOS, it literally stands for save our ship. And it was broadcast when ships were sinking. So that's what an SOS call is. That's what this is named for. This is it's a really bad job of repairing even. So we're taking severely damaged DNA and we're trying to just piece together any nucleotide sequence at this point. And SOS repair is very frequently going to result in major mutations that persist within the DNA long term. All right, this is where we want to stop for lecture video A. We will pick up as we delve into transcription in gene expression, and then in lecture video C, we'll look at translation.